Good evening. I'm Rick Lifton, the president of Rockefeller University, and I am so delighted to welcome everyone to the 20th annual presentation of the Pearl Meister Greengard Prize. This is always one of the highlights of the Rockefeller academic year in which we recognize women scientists who advanced science and helped to shape modern biomedical research. It's great to see so many of you in the audience today, and it's a special welcome to everyone who is joining us virtually this evening. For those of you who might be with us for the first time, please allow me to share a little bit of background. Rockefeller University is one of the world's great biomedical research institutions. We have a singular focus, science for the benefit of humanity. The university was founded in 1901 by John D. Rockefeller, who recognized that the major limit to preventing or treating disease was the lack of a fundamental understanding of their primary causes. Many of the most amazing discoveries in the last century in biomedicine have occurred here at Rockefeller, including the discovery that DNA is the chemical of heredity. In our 122-year history of our 256 professors, 26 of them have won the Nobel Prize in fields of medicine or chemistry. Among our Nobel laureates is the late Paul Greengard, a pioneer in the field of neuroscience and the co-founder of the Pearl Meister Greengard Prize. Paul received in the year 2000 the Nobel Prize in Physiology or Medicine for his crucial discoveries about the mechanisms of signal transduction in the brain that regulates the activity of neurons. His work has shed light on the causes of neurologic and psychiatric diseases, including Parkinson's disease, depression, schizophrenia, and Alzheimer's disease. Paul and his wife, the renowned sculptor Ursula von Rydingsvard, established this prize with his monetary share of his Nobel Award and named it after Paul's mother, Pearl Meister Greengard, who died giving birth to him. They were guided by their prescient recognition that women scientists were not being appropriately recognized for their contributions to science, and they set out to change this. Paul and Ursula's vision and generosity inspired friends of the university to add their own support to this prize, which includes a $100,000 honorarium. To date, there have been 24 recipients of the prize. These recipients have been trailblazers in diverse fields of science, including molecular biology, chemistry, cancer research, immunology, biochemistry, genetics, biophysics, and neuroscience. 3 Pearl Meister Greengard Prize recipients have gone on to win the Nobel Prize. Ursula, who's here with us tonight, thank you so much for being here as we celebrate 20 years of this prize. Would you please stand and be recognized? Thank you. You may have noticed at the reception the Pearl Meister Greengard Prize exhibit in the reception hall. Each plaque displays the name and likeness of a recipient, a summary of her landmark discoveries, and the name of the distinguished presenter of that year's prize. I encourage you to take a close look at it after this evening's ceremony if you haven't already. I'm also pleased to acknowledge my colleagues on the Pearl Meister Greengard Prize Selection Committee, many of whom are joining us here tonight. We're fortunate to have such a distinguished group of scientific leaders taking part in the selection process, including 3 Nobel laureates. The complete committee list can be found in your program. Also joining us this evening are 2 of my presidential predecessors, Torsten Wiesel and Mark Tessier-Levine. It's an honor and a thrill for us to welcome you back here on campus this evening for this milestone occasion. In this 20th anniversary of the prize, we're recognizing 2 scientists who have made groundbreaking contributions to our understanding of the nervous system, Jan and Eve Marder. Lily is recognized for characterizing the first potassium channel and for many other impactful discoveries that you will hear more about later. Eve is celebrated for her work on understanding the modulation of neural networks and demonstrating how they can be both flexible and resilient. Both are iconic scientists, legends in their own time, and committed mentors to the next generation of neuroscientists. I'm also delighted to extend a special welcome to our recipients' family members who are with us in the audience this evening. Joining Lily is her husband and collaborator, Yunung Jan, and their son, Max. Thank you for coming this evening. Congratulations. And Eve is joined by her husband, Arthur Wingfield, a professor of psychology and neuroscience at Brandeis. Thank you for being here this evening as well. Part of the tradition of this prize is that we invite a distinguished woman from outside academic science to present the award. I'm now delighted to introduce our special guest speaker, Ellen Futter. Ellen is a celebrated cultural leader here in New York. She was president of the American Museum of Natural History for 30 years and now serves as an emerita of this treasured institution. New Yorkers know her for modernizing and expanding this beloved museum with the addition of the Rose Center for Earth and Space and the Richard Gilder Center for Science, Education, and Innovation. Ellen was the first woman to lead a major cultural institution in New York City. She led the museum through many firsts during her tenure, including awarding the museum's first PhD degree and the launch of the first freestanding museum-based Master of Arts program in teaching, focused on earth science. A native New Yorker, Ellen has had many careers. Prior to the American Museum of Natural History, she was the president of Barnard College, taking on the role at the ripe age of 30, the youngest person to be appointed president of a major American college. She's also worked as a corporate attorney and was the first woman to chair the Federal Reserve Bank of New York. Ellen is a nationally recognized and highly regarded expert on science education. She has served as a commissioner on the Carnegie Institute for Advanced Study Commission on Mathematics and Science Education and testified before the House Committee on Science and Technology on the importance of STEM education. Ellen has been recognized for her leadership with numerous awards and honorary degrees, most recently the 2022 Medal of Excellence from her alma mater, Columbia School of Law. We're honored to have Ellen with us today. Please join me in welcoming her to the stage. Thank you.
Thank you so much, Rick, for that very generous introduction, though as someone once said, don't be humble, you're not that great. And a warm welcome to everyone this evening. It is really a pleasure to be here with all of you. And I also want to offer my thanks to the selection committee for the honor of being asked to participate tonight, and to my many friends from the Rockefeller Museum of Natural History and Barnard communities who are sprinkled throughout the room, and it's very nice to have all of you here. It is such a pleasure to be here at an institution that I have long admired and to participate in this important event celebrating two distinguished women scientists and their achievements in biomedical research, Dr. Lily Jan and Dr. Eve Marder, to whom I offer my warm congratulations. Well done. And how could I not be inspired by the genesis of this award, which was established by the late and renowned Dr. Paul Greengard, as Rick has already shared with you, and his wife, the internationally acclaimed sculptor Ursula von Rydingsvard, who I was so pleased to meet tonight as well. And the background of contributing his Nobel earnings and naming the award for his mother, who died when he was during childbirth with him, I have rarely seen a more touching constellation of circumstances anywhere. I've had the great pleasure and good fortune of spending the last 4+ decades representing and advancing two institutions that are near and dear to my heart, and whose connections to the work of this institution and tonight's awardees is noteworthy. First, at Barnard, for the advancement of women generally, with a particular focus and strong record on producing women scientists and doctors, and then at the American Museum of Natural History, which is itself an institution of scientific research, and I'm pleased to say is making great strides on behalf of women scientists, including having a woman, Dr. Cheryl Hayashi, who I am delighted is here tonight, who not only is a curator, but also the Leon Hess Director of Comparative Biology Research at the museum, as well as a Senior Vice President and the Provost of Science. And I'm also pleased that we have made of the last 10 curatorial scientific appointments at the museum for tenure or tenure-track positions, 7 have been outstanding women. So thank you. So I know a thing or two about the pace, dare I say, too often slow pace, of women's progress in science. Some of you may recall the famous cartoon that showed the descent of man as a series of footprints, beginning with Australopithecus and ending with a high heel. In Museum of Natural History terms, we think of this as evolution. Slow but steady, unless there is an extinction event, but we won't go there tonight. I should also note that I am far more comfortable talking about these institutions and their important missions than I am about myself. Yet it is the latter that I have been asked to discuss tonight, so I shall try to oblige. By way of confession, mine has been an unexpected, unpredictable, and nonlinear career path, which began, as Rick indicated, with practicing law at Milbank, where I'm happy to note I had the very great pleasure of working on a number of matters for Rockefeller University. So in some sense, this is a return to my earliest professional days. Then things went in a very different direction, launching what my 2 now grown daughters have long characterized as my not a career. As in, although the museum is a scientific institution, I am not a scientist. And while Barnard is an academic institution, I am not an academic. So I am Exhibit A of the fact that life and careers, as I regularly advised Barnard students, can and often do proceed in unanticipated, even unimaginable directions. The trick is to be open and willing to chart new paths and to go through doors of opportunity when they are open to you, which sounds straightforward enough, but actually can be daunting, especially if, as was often the case for me, you haven't actually been traditionally or fully trained or prepared for the role that you are taking on. And when you are keenly aware that by breaking boundaries as a woman and being the first to assume a particular leadership position, you are at once helping to push the doors ajar for generations of women to follow, but also carry the burden of doing well on their behalf. Still, notwithstanding my not a professional foundation, I did have important personal connections to both of the institutions I was privileged to lead, and strong alignment with their missions. At Barnard, I was not only a graduate of the college, but one of legions of alumni who would say that in many ways, Barnard made me who I am today. And I also was deeply committed to the intellectual and personal development of young women and the advancement of their opportunities. And at the museum, I was one of millions who came as a child and marveled at its wonders. And beyond that, while not a scientist, I have long been a naturalist, blissfully collecting shells, which I still do, rocks and butterflies, and developing an early passion for the environment and natural world, as well as for diverse human cultures. And with the benefit of my legal training, I greatly enjoy making the case on behalf of these important, iconic, and both global and utterly New York institutions. While at Barnard, That meant making the case not only for the advancement of women, but demonstrating why the advancement of women isn't just for their benefit, but for the benefit of society at large, to have access to the fullest range and extent of talent possible, meaning both men and women, and that it is also, importantly, better for families and children, as the mother's level of education has long been shown to be important to the health and well-being of children. And more recently, making the case for and leveraging the museum's vast scientific and educational capacity to enhance the public understanding of science, while also opening doors for women and others not traditionally engaged in the scientific arena. And because we are here tonight at Rockefeller, I want to pause on the public understanding and appreciation of science. Because we live in a time of unprecedented knowledge about what it means to be human, about the interdependence of natural systems and species, and, as tonight's honorees brilliantly demonstrate, the mysteries of molecules, genomes, proteins, and neurons, and also a bit farther afield of the oceans and the universe beyond. A time of emerging new frontiers, fresh discoveries in biology, medicine, and technology, with thrilling new fields, cross-disciplinary collaboration, new ways of working, exploring, studying, and discovering. Yet, disturbingly and of urgent concern, there is also a growing resistance to and politicization of science, of expertise, of raw evidence and data, of the scientific process, of truth itself. What an odd and deeply troubling juxtaposition when scientific advancements have long improved human health and the quality of life, as it continues to do so palpably now in combating the pandemic and in so many other areas, including those pursued by Dr. Greengard himself, such as Alzheimer's disease, and of course in other areas such as the moonshot effort to cure cancer and those to mitigate climate change and species and habitat loss. Science has also driven America's economic and global leadership, and it is absolutely the case that science underpins many of the most important topics of our time and likely of any time, as new discoveries and challenges constantly emerge. Think AI, with all of its truly extraordinary potential and promise, but also daunting threats and issues of ethical concern. Still, put simply, science offers many of the most promising directions for addressing society's most pressing needs. So, educating the public, not only about science itself, but about why people should trust it, and about the vital nexus between science and society, is more urgent than ever. As Rick just alluded to, think science for the benefit of humanity. In a way, scientists, like those we honor tonight, are a kind of intellectual first responder, if you will, seeking answers where others often don't know to look, finding and testing clues, and then, yes, offering solutions and advising the public about them. What could be more noble, more civic-minded, more important? Which is why I feel so fortunate to have been able to advance these very pursuits of science and education and the advancement of women, including women scientists, and why I am so honored to participate in this event at this exceptional university, recognizing two brilliant women scientists. Thank you.
Thank you so much, Ellen, for those inspiring words. I now would like to pay tribute to our honorees, starting with Lily Jan. Lily is the Jack and Dolores Lang Professor of Physiology and Biophysics at the University of California, San Francisco. She's also an investigator of the Howard Hughes Medical Institute. The story of Lily's science encompasses physiology, cell biology, biochemistry, fly genetics, and neuroscience. She's probed how ion channels work, identified their molecular subunits, and studied how they regulate nerve cell communication. She has also identified determinants of neural cell development and the mechanisms of diverse dendritic morphologies of nerve cells. Lily has worked with some of the great names in the history of molecular biology. Max Delbruck, who led the migration of physicists into biology in search of fundamental principles of life science, and behavioral geneticist Seymour Benzer, as well as Steve Kuffler, who is often called the father of modern neuroscience. As an undergraduate at the National Taiwan University, Lily studied theoretical high-energy physics. Physics was one of the most desirable majors in the 1960s in Taiwan, and NTU was the best university. Only the best students were accepted into the major, and Lily was one of them. And her future husband and scientific partner, Yunung Jan, was another. Both Lily and Yunung then went to Caltech for graduate work in physics, but they fell in love with biology. After 2 years, they switched fields, completing their graduate studies under the mentorship of Delbruck, himself, as noted, a physicist turned biologist. Lily followed her graduate work in membrane biology with postdoctoral research with Seymour Benzer, also at Caltech. She was captivated by the promise of fruit fly genetics for studying neural signaling and the function of the nervous system. Working with Yunung, Lily developed a technique for recording nerve impulses at the junction between nerves and muscles. They meticulously screened and recorded the behavior of mutant Drosophila larvae, looking for abnormal synaptic transmission. They traced the curious leg-rattling behavior of one mutant, called Shaker, to a defective potassium channel. At that moment, while other ion channels, such as sodium channels and calcium channels, had been purified and their activities characterized, efforts to do this for potassium channels had run into a brick wall. At this stage in their careers, Lily and Yunung decided if they were going to pursue neuroscience, they better learn how to record from what Lily called the tiny little normal-sized neurons of fruit flies, rather than the relatively large muscle cells found in the fly. They opted, therefore, for a second postdoctoral stint, this time with Steve Kuffler at Harvard. And while there, they made their first big discovery, that molecules known as, that simple peptide molecules can themselves act as neurotransmitters, transferring messages from one neuron to another. They further demonstrated that a peptide could transmit messages to relatively distant neurons without synaptic contact, behaving like hormones. They continued their work on neurotransmitters when they returned to the West Coast in 1979 to set up their own laboratory at the University of California, San Francisco. Here they also restarted their study of the Shaker fly mutant. But how to proceed? At that time, the idea of using genetics to identify the chromosomal location of a mutant gene and then using recombinant DNA technology to clone the chromosomal DNA encoding the mutant gene was an idea that had been developed but had not been reduced to practice. The work was arduous. There were no genetic maps. Cloning DNA segments was very challenging. Finding something that was where you wanted it to be in the genome was even more challenging. But they nonetheless succeeded in localizing the Shaker gene by identifying chromosome rearrangements that caused Shaker mutations, and showed that all of these mutants had one end in the same chromosome location, pinpointing the location of the Shaker gene. They then managed to build a DNA map of that chromosome segment and identified the genes encoded in that interval. Again, extremely time-consuming task, and ultimately identified a novel gene that was broken by each of the Shaker mutations. This took 6 years, and you might imagine that the university was very patient to not just recognize the work, but to actually promote them both to tenure during this period. They clearly recognized talent when they saw it. So the shaker gene, was the first known potassium channel, and this discovery led to a slew of high-impact papers, including the discovery a year later of the first mammalian potassium channel, which they showed was stunningly similar to the one they found in fruit flies, suggesting the commonality of the fundamental structure of many potassium channels, which has held up very strongly over the years. The Jans have gone on to study whole families of potassium and other types of channels, defining their roles in disorders as varied as epilepsy and diabetes. They were among the first to recognize that the cell biology, not just the neurophysiology of channels, is important, describing processes like channel localization to different domains of the cell. They've also made seminal discoveries about how neurons develop and acquire specific identities. In recent work, they have discovered a new family of mechanosensitive channels and have been characterizing their biological roles, which include functions in the development of dendrites. At the same time, a parallel thread of work in the fruit fly led the Jans to make seminal developmental insights about neurogenesis, the process by which new neurons are formed, and neuronal cell fate determination. What drives Lily Jan is also one of the most fundamental expressions of curiosity. How does it work? What's the mechanism? Her entire body of research, which we honor today with the Pearl Meister Greengard Prize, demonstrates Lily's remarkable ability to pursue impactful answers. Congratulations. And now it's my great pleasure to celebrate Eve Marder. Eve is the Victor and Gwendolyn Beinfield Professor at Brandeis University, where she has been on the faculty since 1978 and where she was also an undergraduate. Eve studies how neural circuits arise from the intrinsic properties of individual neurons and their synaptic connections. Her work has demonstrated that neuronal circuits are not hardwired, but are flexible, adapting to changing conditions, and can produce a variety of outputs or behaviors. Eve was one of the first neuroscientists to use computational biology interfaces to characterize the nervous system. Her sophisticated combination of experimental and theoretical neuroscience led to remarkable findings about homeostasis in the nervous system, contributing to the current understanding of the brain as being dynamic, balancing flexibility and stability. As an undergraduate in the late 1960s, Eve was inclined to study civil rights law until a course on abnormal psychology put her on a path to biology and neuroscience. Her first encounter with the lobster stomatogastric ganglion, the system in which she made many of her most important discoveries, came as a graduate student in Alan Selverston's lab at the University of California, San Diego. The lobster STG is a collection of only 30 neurons that control complex feeding rhythms in the GI system. Her thesis focused on identifying the neuromodulators, small secreted signaling molecules made by different neurons in the STG, and she was intensely interested in understanding the organizational rules by which these transmitters worked and organized the behavior of the system. She tried 15 different neuromodulators and found that each one influenced the activity pattern in the STG, but in different ways. This was her first inkling that the same neural circuit could produce many outputs and that neuromodulators can create variability and promote resilience and stability. She dug deeper into this work when she returned to Brandeis as an assistant professor, publishing mechanistic descriptions of how neuromodulars could alter circuit behavior. We're all familiar with the names of some of the key neuromodulators she implicated that also affect human emotional states: serotonin, dopamine, noradrenaline, and endorphins, to name a few. Eve's work on understanding how neuromodulators function, creating circuit flexibility, stability, and resilience, also helps to explain disordered modulatory environments that can give rise to disorders such as depression, PTSD, and schizophrenia in humans. In the 1990s, frustrated by the limitations of computer models to depict a biophysically realistic model of an STG neuron, Marder embarked on a productive collaboration with Larry Abbott, then a professor of particle physics at Brandeis. Together, their teams developed self-tuning homeostatic neuronal models that could identify how circuits can adapt to changing circumstances. They also developed the dynamic clamp, a tool that connects a non-biological electrical circuit into a biological neural circuit, a fast, precisely defined brain-machine interface. Later, Eve became interested in the concept of homeostasis in the nervous system. She found that neural circuits will return to a baseline rate of activity or homeostatic goal following a change or disturbance that alters their activity. This led to additional work on the resilience of neural circuits and a surprising demonstration of the impact of climate change. In experiments in which Eve and her team tested the resilience of neural circuits in crabs as water temperature rose, they found that there was a critical limit at which these normally resilient circuits simply crashed. This work, like much of Eve Marder's research, has opened new areas of neuroscience and changed how we fundamentally think about neural systems. Eve believes that the pursuit of science should be coupled with service to the scientific community. Among many roles she has played during her career, she's been president of the Society for Neuroscience, a scientific society that boasts 36,000 members, and served on the working group for the Obama administration's Brain Initiative. She's also a dedicated mentor who has pushed for diversity, equity, and inclusion at all levels of science, efforts that would resonate with her undergraduate self's interest in civil rights law. We're thrilled to honor Lily Jan and Eve Marder with the Pearl Meister Greengard Prize in recognition of their extraordinary contributions to science. Please join me now in welcoming them to the stage. Okay, so why don't you stand here, and you stand here, two stand next to one another, and you stand here and just slide in. Okay, so congratulations. I would like to briefly read from the citation that is being presented to tonight's recipients. Lily Jan, you're being honored for your profound contributions to how ion channels work and regulate nerve cell communication. You've combined electrophysiology and genetics to identify and characterize the first potassium channels, key regulators of neuronal activity, and discovered how peptides can act as neurotransmitters. Eve Marder, you're being honored for your foundational research on the properties of neuronal networks. Your research has been instrumental in demonstrating that neuronal circuits are not hardwired, but are dynamic, able to balance the need for both flexibility and stability. And Ellen, would you now present the citation?
I'd be delighted. It is my great honor to present Dr. Lily Jan and Dr. Eve Marder the 2023 Pearl Meister Greengard Prize. We have it. An international award recognizing outstanding women in biomedical research, and on behalf of the selection committee and everyone here, congratulations to you both. Okay, let's take some pictures. All right.
So a few photos. Thanks. I think we're done. Fastest photographer. Thank you. Okay. So please come over and have a seat. Well, again, congratulations, and we're thrilled to have you here with us tonight. We didn't want to pass the opportunity to have a discussion with you. Maybe start by just asking, in your careers, obviously neuroscience has come a long way, in no small part due to your work, but be interested in your take on how neuroscience has changed over the course of your careers, and particularly how the role of women has changed.
I think neuroscience has both changed a lot and not very much. I think some of the core questions that were around in my early days are still there. I mean, I think we all still want to understand how circuit function arises from its component parts. We have tools that were unthinkable 50 years ago, and those tools are bringing all sorts of new insights, but the core problems and the core principles, I think, are still largely there. I mean, maybe you would disagree.
I agree. The questions that interested neuroscientists or students way back when we were young to now are in a way, obvious, interesting questions. It kind of reminded me when we were in Seymour Benser's lab. You know, we had this lunchtime that lasts for a very long time. And we were not really modest, you know, the students and postdocs in Seymour's lab. We went to a blackboard and just wrote the questions that each of us were interested in. But at that time, there was really not obvious at all that those questions can be addressed, but they are being addressed now.
So, from the time you started as graduate students, how have you seen the role of women evolve in neuroscience and science more broadly?
Well, when I was a graduate student, there weren't any women faculty. So it was very simple. I mean, you know, students today always talk about role models, and I sort of laugh because all the women I knew who were doing science were working in their husbands' labs. I mean, so that was a very respectable thing to do, and I didn't actually even know there was another path. I think there were always really smart women doing really wonderful science. I knew a few others in other fields, but I personally was always very grateful to be given an opportunity to do science. And so I think the thing that has changed from then to now is we didn't think about mentors, we thought about people who gave us opportunities. And I still think while I try and be a good mentor, I think the most important thing that we can do is to provide opportunities to talented, determined people.
Right. Agreed, because we had the same observation and experience. And way back, women were interested in science, but they got much better opportunities with everybody's awareness and effort.
So, Lily, you had the relatively unusual role of having an explicit collaboration throughout your career with your husband, Yunong. How did that come to pass, and how has it worked, and how did you divide up activities? And interested in your take on it.
Well, I guess just naturally you'll be interested in your friends and your spouse work in science. When we were graduate students, Max made sure we were not doing anything similar on our thesis work. It got started when we were taking the neurobiology course at Cold Spring Harbor, just to get ready for postdoc. That's in a way by chance, but it worked out really well.
So, and Eve, you had an interesting collaboration, probably one of the first collaborations of neuroscience and physicists with Larry Abbott, who is here with us tonight. How did that collaboration come to pass?
We both had graduate students who had the same fellowship, and they were taken to a lunch and they started talking, and his graduate student, Tom Kepler, said he was working on memory, and my graduate student said, how can you be working on memory if you have nothing to do with neurons? And so they started talking, and then Tom brought Larry over to see one of Jim Wyman's, Mike Nussbaum's experiments, and they threw my poor postdoc lost his preparation as the two of them started playing with the knobs and dials. And I think it was a case where having physicists who are really interested in ideas about neuronal activity and memory could actually see a real nervous system in action. It triggered everything. And then Larry and I just got along really well. I sometimes say he was the best student I've ever had. And he's done okay, you know. He actually did something which I think is really remarkable, and if there are any physicists in the room who want to become neuroscientists, you should do what he did, which is he actually read every day. He read all of the Kandel, Schwartz, and Jessel book from start to finish, and he'd come and ask me questions. And so he really learned neuroscience from the ground up.
So Lily, Max Delbruck was famous for exhorting young scientists to not do fashionable science. Did that influence your selection of problems, and how has that played out for you?
Yes, and we still follow that guidance. It, in a way, makes sense. I was going to quote Feynman, but I couldn't quote it verbatim, but you can look it up. But same idea that if many people are working on the same problem, how much more can you do? And if there's something you are really interested in, go for it.
Eve, I suspect the STG was probably not the most common source.
Yeah, at the time it was actually it was more mainstream then in a sense than it is today for a variety of reasons. But I would just like to resonate. I didn't have anybody say that to me, but I remember as a graduate student, the big in thing was to try and purify the nicotinic acetylcholine receptor. And I said, oh, it'd be really exciting to be part of a team to do that. And then I said, but it's going to get done whether I do it or not, so why should I bother? You know, I can read about it. And so I actually figured out for myself that I always wanted to do something where my participation would change something. So I came to the same conclusion. So I never knew that Lily and I were sitting with exactly the same conclusion, but I thought if I was going to do something, it should be something that no one else was going to do quite that way.
Terrific. I also was taken by your selection of the STG circuits as from one of Einstein's oft quoted and plausibly real quotes of reduce a problem to its simplest dimensions, but no simpler. And the STG system seems to fulfill many of those requirements.
Yeah, no. It's like this. When I started, people called systems like the STG simple systems. I never say it's simple now. It's just small. And so I think what's really wonderful about the STG as a preparation is it's small enough so you can get your head around and your experiments around identified neurons and define problems, but it is not simple. If it were simple, I would be out of work, right? And we would have done it, but there's still so many unbelievably fascinating problems that because we understand so much, that we can still envision understanding more.
So we have a number of trainees at various stages of their development in the audience tonight. What advice would you offer them in picking problems and career paths?
We were talking about that a little bit at lunch. It really is a long game we are in, and go with the question that really interests you.
I think, just to reiterate what Lily said, I mean, my version of that is to say that doing science is sufficiently difficult but the only thing you should work on is something that really excites you. Because if you're working on something that doesn't excite you, then it's just hard. And if you're working on something that's exciting, then it's hard and really exciting.
That's great. We have time to take a few questions from the audience. If you have a question, please raise your hand and a microphone will find its way to you. So a question on the aisle, halfway down here.
There's one in the back as well.
Okay, please go ahead.
Hello, congratulations, guys. Congratulations, Eve. What do you guys have to say about the idea that consciousness precedes matter?
Nothing. That's beyond my limit. Do you have anything to say?
There's a question down here on the aisle.
Hi. Yeah, I have two questions, actually, which is a little bit annoying. But the first is that something that I find really impressive about both of your work over the last several decades has been the transcendence across fields, right? That, sure, you've both worked in neuroscience, but you've gone from very, very, very specific, like chemistry and the phenomena of, monoamines and stuff like that, up to circuit dynamics and systems and development of nervous systems and things of all sorts of scales. And when I read neuroscience papers now, I feel like there's a much more tunnel vision in the scope of each of the individual papers. And I'm wondering how you keep the holism in mind about how to think about the function of the nervous system across all these scales, across time scales, spatial scales, and everything like that, and use that to guide your research while still publishing papers that are digestible and interpretable. And then the second question is that I found it a little bit striking that all of the quotes and references and foils and contexts that have been provided for you guys today have been men, and I'm wondering if you could speak to one or two women in your lives who may have been inspirational figures as well.
I'll take the last first, and that is when I first started at Brandeis, Kalpana White was on the faculty. She had been appointed the year before me, and she said a few things over the years when we were very good friends. And one of the things that she said, and I think this is deeply right, she said that if you keep studying a problem, eventually you'll find gold. And I think what she meant by that is there's so much beauty and richness and mystery in biological problems that if you just keep going and keep your eyes open and your ears open, you'll find something that no one's ever seen before. And so that was someone who was just a year ahead of me. I mean, I didn't know very many more senior women who had anything to say. And I think over the years, Kalpana we would have these conversations. And so I think most of the women who were the most important were either my peers or my trainees. And I've learned a tremendous amount from really smart trainees who've had really important insights.
I totally agree. Actually, we look like mentors, but we learn a lot from our colleagues in the lab. Yeah. It's really an exchange. Yeah.
And then the question about working across many scales in the nervous system.
Right. So I think it's almost a little bit of an embarrassment of the riches nowadays that you could actually pick any scale and stay there for a very long time. And so it's really what you want to do. You could do that, or you get surprised from whatever you're doing and then go up or down just because you are curious about that.
Question down here.
Thank you, Lily and Eve. It's such an honor to hear two outstanding women scientists talk about their life in science. Just the other day, there were a group of women at Rockefeller, scientists, trustees, and we were all thinking about the word everyone is talking about AI these days. And as we have the opportunity tonight to talk to two outstanding women scientists, I think I'd like to ask you, how do you think, as we use AI to establish new models of thinking and inquiry, how do we make sure that if there is such a thing, women's way of thinking and women's values, women's empathy, is captured in these models that allow us to explore further?
Oh, that's for you.
I thought you know. Right, so for the computation, you are much
yeah, but
closer.
That's not
right. So for me, it's really kind of an outsider's view about machine learning, AI, and everything. And to me, it felt like they were learning from whatever that's accessible to them, just at a much faster, a more expensive scale, right? And so that goes back to what was it that they were going through. It could be up to this point they are predominantly from one gender versus the other, but that may not always be the case. That's the level that we are involved in. Whatever we put in the literature, we put it, put out as the data that other people can scan and figure out things maybe we cannot, as human beings, pick up so easily.
I mean, I'm not going to answer your question, but I'm going to tell you two things that I've observed lately. One is, at one point I was writing a paper with some trainees, and I said to this person, somebody must have done this experiment in 1986. Find it. And so he came back and he said, no. And I said, I know it was done. So I went and did my own search. 10 minutes, I found the paper, and it was 1985, 1986, but And then this happened several times with different trainees, and I asked them what they were doing when they were doing their search. Well, they were all using Google Scholar, and I was using my brain and PubMed. And I discovered then, I started talking to them, and I discovered that Google Scholar, if you do a search, uses the most cited papers and the most recent things. So it uses AI to give you back the return of the search. And it failed miserably to find the important and salient work that was a little bit older. And today, if you do a Google search on anything, which again is an AI tool, right, you discover that it's actually less useful today than it was 5 years ago. And that's because the learning from the behavior of the aggregate is not always learning the most useful thing. So I don't know. I think this is a really hard problem. Everybody's going to be facing it. I just will tell all the trainees, you cannot use Google Scholar to do useful searches because you won't get the old stuff that's really relevant. And you don't want to send a paper to a journal and have some reviewer send you back, well, this was already done in 1986, so why didn't you cite it? So go find it.
Our following days, as more inquiry is made of how AI should be regulated and guardrailed, because when you see some of the tech leaders who are going to Washington and speaking to the powers that be, so often it's white males. And I think that's going to color the way in which AI is structured and how inquiries are made. So, and the answer is going to be often influenced by how the question is asked. And women do it quite differently than men. Better or worse, but we need to have both sides in the world.
So I think this is a really interesting issue. When I was a beginning assistant professor, there was early in the women in science movements, there were lots of people who said that women did science differently than men. And there was a whole scientific inquiry, and philosophers and gender studies people were arguing about. And I'm not sure, people said, well, women are more likely to be afraid to make mistakes, but there was a whole line of inquiry. I'm not sure that's really true. In other words, I would argue today in 2023 that a really creative scientist is a creative scientist, and a less creative scientist is a less creative scientist, and that there are tons of people who just follow the hordes, who do what I call consensus science, and there are men and women who do consensus science, and there are men and women who do imaginative and creative science. So I'm not sure that that underlying assumption that men and women think about science differently holds. I've heard much less about it recently than we were hearing 30 years ago. So now, whether people should be asking intelligent women who know things about AI, yes, they should. It's not me, because I don't know anything. But I think the really interesting question about whether men and women really think about science differently, I would say it's an open question.
Well, we're at time, so I want to thank the audience, but also one more time.